Okay, I thought it was going to be a horrible roast. Well, thanks, Graham, and I'm really honored to receive this award, and I really want to thank Brett and the voting committee for giving it to me. It's a real pleasure and honor. As Graham noted, I do love collaborating, and over the years I've been involved with many projects, and I was thinking about what to put together, and it's difficult. It's almost like telling one of your kids, you love them more than one of the others. So what I decided to do is pick the one that keeps me up late at night, and that is the C9orf72 project. So I'm going to tell you, sort of highlight the work that we've done on C9 for two CNS diseases. One that Alex McCampbell talked about yesterday, which is amyotrophic lateral sclerosis, ALS, and frontotemporal dementia, which are oftenly found together. So the excitement in C9 essentially started in September of 2011 with two seminal publications, one from Rosa Rademacher's lab over here, and the other one from Brian Trainor's lab, both documenting the presence of this hexanucleotide repeat expansion here in a gene with a really weird name called C9orf72. So essentially, the most important thing about this discovery was that it actually explained the majority of cases in ALS and FTD with a predominance that was incredibly high. So let me orient you a little bit on the C9orf72 gene. It's actually quite complicated. So it's up here. It has two first exons, exon 1A and exon 1B, and then all the exons downstream. It has 11 total. The ATG is in the second exon, and the stop codon is in the last exon. And the expansion, which is found only on one allele in patients with ALS and FTD, can be thousands of repeats long. And that expansion is found between the first two exons. So the fact that this expansion is only found on one allele sounds a lot like the triplet repeat diseases and really had the flavor of a toxic gain-of-function mechanism of disease. So yesterday night, for fun, I actually went into PubMed and just typed in C9orf72, and from 2011 up to today, it's been 600 publications. So it's just amazing how much publishing has been going on. And so all those publications have resulted in three potential mechanisms of disease. So one of them is a haploinsufficiency. So this mechanism postulates that the repeat expansion, which is upstream of exon 1B, leads to its silencing. And because the expansion is only found on one allele, that would lead to a silencing of that one allele and a haploinsufficiency, a 50% reduced protein. And this is some of the supporting data showing a 50% reduction in the expression of the mRNA produced from 1B. The other mechanism is by the fact that the transcripts that initiate at 1A will produce RNA that have the expansion, and this is in the sense direction, and then will produce RNA structures as shown here, and these will then bind RNA binding proteins, titrate them out from the nucleoplasm, and then result in changes in gene expression. And this is sort of the mechanism that Charles Thornton was talking about the other day for myotonic dystrophy. A third mechanism of disease that many labs have now documented is that these RNAs, the ones that escape into the cytoplasm, can actually be translated. But they're not translated in the canonical way using an ATG that is upstream, because there's actually no ATGs found upstream of the repeat expansion. So this mechanism was initially discovered by Laura Raynham. It's called RAN. Probably Laura coined it because of, she wanted to become popular. But it essentially stands for repeat associated non-ATG translation. And the reading frame of of this repeat is such that you can produce 3 peptides, either GA, GP, or GR. And that's what's shown here. And these peptides tend to aggregate and are found in the cytoplasm of neurons in human patient material. So I think that, probably it's going to take another 10 to 15 years to fully understand what actually causes C9orf72, but I think that we can all agree that if we had that what we should do is if we had a strategy to reduce the RNA toxicity or the dipeptide repeat protein toxicity, it would be an on-target disease mechanism therapy for C9, and if we could target the whole central nervous system, that would probably be beneficial for patients with ALS and FTD. So before I get to the oligo work, I just want to touch on some mechanistic work that we've done, trying to, in our hands, sort of dissect the disease mechanisms. And before I show you this experiment, what we did before this is we developed antisense oligonucleotides to target mouse C9orf72, and we injected these into the ventricle of mice, and that resulted in a 2 to 3 month knockdown down to 75% or so of C9orf72, and we were able to find no adverse effects on mouse behavior. These mice were perfectly fine, they had motor strength, no obvious phenotypes. But that was only a partial suppression of C9. So now we decided to go and generate a genetic model where we eliminated the rodent C9orf72. So we essentially just deleted exons 2 through 6 of the mouse gene by inserting a LAG-Z cassette. And you can see here, as expected, the RNA of C9orf72 in the heterozygous is depleted by 50%, and then the knockout, it's almost gone. And this is also mirrored when you look at C9orf72 proteins. So you get 50% and then gone in the knockout. So what happened to the survival? So in the heads, essentially the survival was normal. There was 1 mice that died due to fighting in the cage. So that's positive. And in the knockouts, the mice were born normally, so there's no developmental defect. They lived up to 200 days, and then after 200 days, we started noting that about 20% of the mice died. And we are still trying to understand the cause of death. The things that we've noticed so far is that the spleens of these animals are quite large, and there's evidence for a T-cell lymphoma. So that's sort of the peripheral issues that this model has. The good news is that we've looked carefully whether there's any CNS phenotypes, and we can't find absolutely anything. The motor neurons are normal, these mice have normal strength, there's no microgliosis, astrocytosis, so we don't think that these mice actually have a CNS phenotype. So we're not too much in favor of the haploinsufficiency model as the only factor that explains the disease. So for that reason, we decided to embark on generating C9 transgenic mice. And the back that we used to do that is shown here. It has exons 1A and 1B, the expansion, and then the back is truncated slightly after exon 5. So this graph over here just shows the expression of C9, the human transgenic, in a variety of lines, each denoted by a bar over here. So as you can see, there's many mice that have single integrations of the transgene, and there's mice that have double integrations. And the nice thing is that we have mice that have similar expression levels with different sizes of repeats. So we started characterizing a few of these lines. The first question we asked is, do they have foci? Do they have RNA foci that accumulate? And so the answer is yes. So we did fluorescence in-situ hybridization using a probe that targets the repeat, and that's this data. And we see RNA foci in hippocampal neurons, as you can see here in the nuclei, and also in spinal cord motor neurons. So over here is the quantitation when we looked at 100 nuclei. The line that has 110 repeats actually has no foci, and the line that has 450 repeats has about 200 foci or so. So we wondered, why does this line have no foci? And so we looked at the expression level of these two lines, and the 450 repeat line has about fourfold higher expression than normal mouse C9orf72, and the surprise was that the line with 110 reads has about equal levels of expression. So after all, size actually does matter. Of course, we also wanted to check whether there were dipeptide repeat proteins produced in these mice. And so we obtained antibodies to do staining, either the GA, GP, or GR. And as you can see here, this brown staining shows you the aggregated dipeptides in cortical neurons. So some of them are cytoplasmic, and then other ones are more nuclear localized. This is just the quantitation of those aggregates. There's anywhere from 10 to 30% of cells have either GA or GP aggregates. So, and then of course, we looked at the phenotype of these mice. And the bad news is that when we looked for any ALS phenotypes, they were absent. So these mice had no impaired muscle function, there were no ALS phenotypes. So then we looked to see whether they had any phenotypes of FTD, and here we were pleased. So for the interest of time, I'm not going to go into explain exactly how these maze experiments are done, but essentially what it comes down to is that there's a series of mazes, and there's either an exit or there's an exit in this paradigm, or there's a reward. And essentially you score the times the mice fails to either exit or get the reward. And essentially that's what's shown here, the number of errors. So we looked at this in two lines with 450 repeats at different times. And so I'm just going to focus on this graph, which shows that wild-type mice over time make less errors, but the transgenic mice keep making errors in finding the exit platform. And you get a similar result when you look at it in a different paradigm. Okay, so this is where the oligo work starts. We wanted to see whether we can develop a oligo that would specifically attack the pathological features of the disease, which is the foci and the peptides. So we did a lot of screening. We screened many oligos upstream of the expansion. And we used here MOGAPmers, 20 mers. We did a lot of screening. And so in this particular experiment, we are transfecting patient fibroblasts with 1,000 repeats and with a lipid transfection. The control oligo does not lower the expression by real-time PCR of isoform 2. This is the isoform that starts transcription at 1A. But then these two oligos, which are upstream of the expansion, have a nice knockdown of C9orf72. The most important part is that these two oligos don't seem to lower the levels of total C9orf72 when these are measured with a primer probe set to exon 2. So we're really getting knockdown of the transcripts that start at 1A and that contain the expansion, and we're leaving intact the transcripts that start at 1B. So does this work in mice? And so we got the transgenic mice, we did an ICV injection into the brain, waited 4 weeks, and then analyzed the RNA for knockdown. The most important here is the oligo in green, which is slightly upstream of the repeat. Don't pay attention to the purple. So here we're looking at the RNA produced from exon 1A. You see a nice knockdown in the cortex and spinal cord. Then we looked at the levels of total C9orf72. There's a little bit of knockdown, about 20%. But this knockdown is probably occurring because when you measure total C9orf72, you're also measuring the transcripts that initiate at 1A. And so as proof of this, we measured the transcripts that initiate at 1B. And as you can see, the oligos didn't really affect transcripts that initiate at 1B. So what about the foci? Are we actually reducing foci in these mice? And so the answer is yes. Oligo 1 caused an about 3-fold reduction in the level of foci in the dentate gyrus. And what about the dipeptide? So gratifyingly, yes, there's a robust reduction of GP and GA in 2 regions of the CNS when GP and GA are measured by ELISA. Okay, so The thing that we're currently working on right now is trying to see if the oligo approach corrected the FTD phenotypes in these mice. So hopefully next time we can report on that. But I hope that today I've convinced you that even though we don't fully understand the mechanism of disease, an ASO therapy towards C9 will minimize the further lowering of C9orf72. As I showed you, the oligos are specific to the transcripts that start at 1A and have the expansion and don't affect the levels of expression of 1B. This approach definitely reduces RNA foci, and as you see, it reduces the levels of RAND peptides. And so I just wanted to put this slide up because it really shows how fast we can move with the Antisense technology that we all share. And so the discovery of C9 happened in 2011 on September the 21st. On September the 22nd, we actually already started working on the disease. Then in 2012, we showed the proof of concept that we can target C9 in cells. Just recently, we've achieved this proof of concept in mice, and we are planning now to start clinical trials, hopefully in 2017. So this, I think, is a testament to how quickly one can use the technology to make a significant impact on disease. And so for the C9 project, there's a tremendous amount of people to thank that are all way smarter than me. There's a tremendous brain power on this slide, all the way from the Cleveland lab to the Ravitz lab to Len Petruccelli at Mayo, the Baylor lab, Jeff Rothstein at Hopkins, of course, all the ISIS family, and Alex McCampbell at Biogen. So just in closing, I obviously, I can't stand here without thanking a huge number of people. And those people are the people who work for me and the people who I work for, like my bosses and my collaborators. And I honestly just simply view myself as the bridge that brings them all together. Thank you.
Thank you, very interesting. There were recently some papers suggesting that nuclear pores were involved in the pathology of this disease. Would you?
Yeah, that's correct. That is a very nice series of papers from Rothstein, Aaron Gittler, and others. And in one of the papers, collaborating with Jeff Rothstein, we're able to show that the nucleocytoplasmic shuttling that is deficient in C9 can be corrected with oligo treatment.
Is this on? Nice talk. So no, I agree. This is an amazingly, fast-moving field, and so we and others do still think that haploinsufficiency does matter. That it's, and I mean, as a general lesson to all of these diseases, right? We probably need to address both alleles in many cases. So, but I was going to ask you. I mean, again, there's several labs have now, for example, found, in a third of these patients, you have methylation of the locus. Do you think that impacts at all on your approaches? And that could be a protective mechanism, incidentally. That's my first question.
Yeah, so I think the methylation will be essentially the mechanism for silencing of transcription from 1B, and it contributes to the haploinsufficiency mechanism, and I totally agree.
Would you stratify based on that, or?
No, so
no?
I think, I mean, if I were to bet, probably haploinsufficiency will play a role. Now, the beauty of our approach is that we're not going to make the haploinsufficiency component any worse, because we are targeting a transcript that is upstream of 1B. So what I didn't mention is that the majority of C9 transcription in both mice and human cells starts at 1B, right. 90% of it. So we're really not touching the majority of C9.
Right. So that's not even, we can talk about that later. Second question, I think, important one is again, most people today, really think that the clogging up the nuclear pores, as the gentleman just asked about, is an important mechanism. And what little we know about that mechanism, it has to, whatever therapeutic we introduce, I think has to be very, very efficient, right? So,
Yeah, so
I was
we
kind of
haven't-
comforted to see that,
Yeah,
that
we
you had a
haven't-
very strong effect on dipeptides.
Yeah, I think that there's enough pores that are okay, that there could be trafficking of, fairly small antisense compounds into the nucleus. So we haven't seen any effects on oligopotency or efficacy in the background of a nuclear pore deficiency.
Well, my question is more simple, right? What degree of knockdown do you really need, to impact on nuclear pore transport? Can you speculate on that?
Yeah, Jeff Rothstein, his knockdown was only about 40 to 50%, and he was able to completely recover the whole deficit with his GFP shuttling assay. So it actually took very little to have an impact on that.
But that's a very artificial assay. We can talk more later.
Yeah, but it's the only thing that, so either I can say we don't know, or we've done an assay that is, what all we have so far.
Okay, next question, please.
Thanks.
The patient fibroblasts don't have that phenotype. You really need to do it in neurons.
Okay, so there's no defect in the nuclear pore?
No.
Frank, congratulations. My question is very simple structural question. Does this DNA exist as a quadruplex and then a duplex? Is there any fundamental structural information known about the gene?
Christopher Pearson has done experiments that indicate that it exists as a quartet, both at the DNA and RNA level. But that, you would think that such a nasty structure would make it difficult for the oligos to actually knock down the target. But that's luckily not what we've seen, that we can do it. And presumably the nuclear exonucleases have evolved to deal with all sorts of complicated structures, because the genome is pilfered with repeats all over the place. Oh, I don't know that. Yeah.
Okay, Andrew, last question.
Thank you very much. I just had two questions about your mouse model. First of all, do you see the stereotypical pathology in the brain that you would see in patients in the mouse? And secondly, do you see the hexanucleotide instability in different tissues? And importantly, is there a change down the generations?
So in terms of the pathological features, of course we have foci and peptides. What we haven't seen so far is the TDP43 pathology in these mice. In terms of the, we also haven't looked still at the age-dependent increase in repeat size in these mice. It's something that we're currently looking at. But the good news is that at least in the germline, the size of the repeat doesn't change over generations.
Okay.
I think that was the three, right?
Thanks.
Okay, let's thank Frank again for a wonderful